Well, it looks like we've got a number of people joining. Give it another minute or so. Okay, good morning, good afternoon. I think we can get started on the meeting this morning. So for an agenda, I sent this out last week. I'll go through just an executive summary of things that have gone on in the past month since our last meeting, and then get into some recent news about some grant applications that we've applied for. And then I want to talk about the in vivo studies that we've recently started, this being the FOP model, some new formulation work that's been done, and the DIPG models that we have upcoming. Next, I want to discuss some of the new data that we have for 2308, that being some hERG and ion channel data, some kinase assay data for ALK2 isoforms, and some new nanobret data for ALK2 mutants with 2308. To finish off, I'll just go through some of the data gaps and next steps for the program. And then I'm going to turn it over to Dan at Charles River, and he's going to give an update on some ALK2 PROTAC work that he's been doing. So just before Christmas, we received notice that we were successful in our application for the Conscious DMOS grant. So, I mean, really, this is a significant milestone for the team to enable us to continue to advance this program into IND enabling studies. So, there were, as expected, a few requested changes and comments to the grant, but we submitted those last week. And I expect we'll be starting the onboarding process fairly soon. We also completed the full application for a Cure Search grant, and the results from this grant are expected the earliest in March, so we'll keep our fingers crossed for that one. At the last meeting, oh, yes.
Pete, sorry to interrupt. I just, the slides don't seem to be moving, and there's a panel that's blocking the slide. I don't know if you can, there you go. That's,
oh, sorry about that.
See if you can get that, see if you can minimize that. Thank you.
That better?
Yes, thank you.
Okay. Yeah, so next, we have started some in vivo studies, and the FOP model done at the University of Pennsylvania, that's nearing completion. And we have some dose tolerability studies underway at the OICR in the UK with 2281 and 2308. Once those studies are completed, we have we'll move those into the queue for DIPG mouse model studies. For that study, previously we had done formulation with 2009 in 0.5% carboxymethylcellulose as a vehicle. When we looked at 2281 and 2308 in that formulation, it wasn't compatible. The 2281 was not soluble. So we did some formulation development work, and we found we were able to solubilize these compounds in a sodium citrate buffer without the need for any additional solubilizing agents or surfactants. Uh, just before Christmas, we received some new data on 2308. We sent 2009 as well because we wanted to make sure we had a benchmark because we were sending this compound to a different CRO for this study. Um, what I can say is that from the data, it doesn't appear that that compound has any cardiac liabilities. Um, we tested 2308 against some ALK isoforms, and it appears to follow along with the SAR for the other compounds in the series. We also ran nanobret for 4 ALK2 mutants, and it performed extremely well in those assays, essentially bottoming out the at about 2 nanomolar in the 4 um, experiments that we ran, and that was done at Reaction Biology. So now I'll discuss the recent grant applications. So just a little more on the DMOS grant. So we did receive notification that we secured that award and submitted all the required changes last week. The co-PIs on this grant are David Euling and at the OICR and Neil Vastev at CAMH. So they brought in matching funds for this award. We also received some pretty good news that they've bumped up the reimbursement for Agora M4K to 50%. And this is really significant because it will allow us to leverage this grant to spend on eligible expenses in Canada. And right now, our thought is to use this grant in combination with the money that we have from the Krembil Institute to support safety and toxicology studies, which there are a few CROs, including Charles River in Canada, that could do that work. For the Cure Search grant, we submitted that last week with OICR as the lead for that grant. And if successful, I think a lot of this grant we're going to put towards CMC, so scale up of the API and stability studies and things of that nature. Just because this grant would give us some more flexibility to engage contract manufacturing organizations outside of Canada. So we're hoping to hear back from the results on that grant sometime in March. We did apply for a small grant through the Brain Tumor Foundation with Ahmed from OICR, but unfortunately we're not successful with that grant. But there is another grant coming up called the Chad Tough Game Changer Grant, and we're looking at applying for that and exploring a joint application with the Jones Lab in the UK. Any comments on grants? If not, I'll move ahead into the in vivo studies. So you'll recall, I think from the last meeting that we discussed this FOP model that's being run at Eileen Shore's lab at the University of Pennsylvania. And we used this model previously with 2009, basically as a test to look at target engagement with ALK2. So we decided to run this experiment again with our 3 lead compounds. We had good efficacy with 2009 at 100 mg per kg. So this time around, we decided to drop that dose to 50 mg per kg for the 3 compounds. There was some extra animals available, so we decided to add 2 additional experiments at a lower dose, 25 mg per kg for 2009 and 2308. So these animals were dosed on the 10th of January, and this is done by Doug Roberts in Eileen's lab. And the final dose was administered on the 24th, so just this past Friday. And the plan was to collect plasma samples and brain samples once these animals were sacrificed. And those samples will be sent to the OICR for analysis. So I'm hoping, we'll see some data once they run through the histology on these animals relatively soon. So early on in the program, we've done a lot of formulation work, in vivo work with using DMSO, PEG 400, and tween as the vehicle. This is fine for some shorter in vivo studies. However, when we ran the longer studies in the DIPG animal models, we saw some toxicity in that using that vehicle. So we switched to 0.5 carboxymethylcellulose for a lot of those studies. However, I mentioned when we tried to formulate 2208, you'll see that there's a large chunk of material that remains. So we were not able to solubilize 2281 in 0.5 CMC. Tried a few experiments to see if we could get this to go, heating it up and sonicating it, but didn't really seem to work. With 2308, we're able to, I would say, get it to solubilize somewhat and have a uniform suspension, but certainly not a clear solution. So with the help of Taira at the OICR, we looked at some different formulations, phosphate buffer. We tried a few surfactants with CMC. That didn't really seem to work overly well, but 150 millimolar sodium citrate, you can see that we were able to solubilize these compounds nicely, and we get a nice clear solution using that. And we didn't need to add in any additional solubilizing agents. So we sent this, the recipe is essentially for this formulation to Rebecca at the ICR, and they're using this formulation in the dose tolerability studies. If Rebecca, you're on the line, perhaps you could just give us a brief update. I believe we're about 2 weeks into the dose tolerability studies.
Yeah, hi everyone. Yes, 2 weeks today. I did touch in with Claire, who's running these assays for us in our animal house, and we've tested 25, 50, and 100 mg per kg, and she was saying that the 100 mg per kg mice looked a little bit agitated and they were kind of over grooming, but the others all looked fine. In the end, we went with 2281 was in the sodium citrate, but we kept 2308 in the methylcellulose because we weren't sure. But at this point, we didn't have much experience of running the sodium citrate. We didn't want to see the tox because of the vehicle. Like you say, it's not clear, but it's homogeneous in the solution. So we've moved forward with the methylcellulose for 2308. We've included some vehicle mice that'll just get the sodium citrate buffer alone, just to check that we're not seeing anything just because of that rather than the drug. But all is looking okay at the minute, and as soon as we have some data, I can forward that to everyone.
Great. So then the next step after that, so these mice will be run for another 2 weeks, and then the plan is to move these into in vivo studies in Maria's lab in Australia, and obviously in-house. Do you have an update on the status of the mice here? And is this going to is essentially everything lined up that once this tolerability study is complete, she can start dosing mice essentially as soon as possible?
Yeah, so Maria in Australia, she's aiming to inject at the end of February, and it will be dosing at the end of March. Here on the slide, it says 2308 or 2281, but I checked with you, Peter, and the decision was to go with 2281 for this study. And then the in-house experiment, is set to start, I think, actually end of March now, beginning of April, just with how things are working out with staff in the animal house. But yeah, this is happening as quickly as we can possibly do it. Yeah.
Okay. Great.
Can I just ask?
Sure.
Can I just ask a question about the tolerability study, the formulations? If I've understood it correctly, we've got 2308 in the meth cell formulation.
Yep.
Is that right? And 2381 in the new sodium citrate buffer. Have I understood that correctly? Is there a risk there we won't get any exposure to 2308 then, given what we saw? And was it soluble? I can't remember. Can we go back to that photo, Peter, which was the one that precipitated in the meth cell?
So we have had drugs previously.
Okay.
That the solutions aren't completely clear, but there's no solid, there's no particles. It's just, and it's, you know, the liquid looks the same throughout, and that's been fine previously. I mean, we'll have these results and PK to go along with it. Hopefully in the next, the tolerability will run for 4 weeks, so 2 more weeks of dosing, and then PK after that. So we should have some idea. I mean, yeah, I get your point. Probably something to note.
Yeah, because there's some extensive, I mean, just for your information for people, there's some extensive literature summaries around formulations, tolerability, and repeat dosing in all species. There's an author called Gadd, and I think it was about 2016, 2018, and they've basically reported out all the toxicology data they could find on a multitude of formulations and excipients. So I've used sodium citrate before, but I couldn't remember. I'd have to go back and check for durations of dosing, and we've used it for the pH control like we've done here as well.
Yeah,
just something to bear in mind. Obviously, we want the best exposures we can to give us the best chance of success.
Yeah, sure. I mean, for the quick model, well, we'll probably be dosing for around 4 to 6 weeks. So hopefully the tolerability that we're running now should cover anything that's going to come up. It won't be a surprise in the efficacy study. But yeah. I mean, is this? Do you think that because if you think that this is an issue and it's something that we should address, it's probably good to discuss this sooner rather than later. Claire unfortunately can't join the meeting, but I could feed back to her that there might be concern about having 2308 in the methylcellulose, if you think.
I think all we'd need is, if we can, a PK profile in the vehicle, if we can get it. Now, I don't know what you're planning on taking at the end of the study, whether that's just limited time points. And off the top of my head, I cannot remember what the vehicle was that was used for the PK studies at Pharma on.
We're going to 2 hours post dosing, I think, is the plan to take the mice for PK, and we look at the brain and plasma.
The vehicle for the PK study at Pharma is this DMSO PEG. And
the original one, okay.
Yeah.
Because what we don't know is if the PK changes at all or in any way between the different formulations. Mm-hmm. So it would be nice to know if we've got similar PK or not, even just after a single dose, not necessarily the repeat dose, because there's other questions arise from repeat dosing in studies, but just to understand the single dose PK in the final vehicle that we're planning on using in critical. If we're going to use these in the efficacy studies, I think we need to confirm our understanding of the PK before we go into those long-term efficacy studies.
Mm-hmm.
And I thought, Rebecca, you mentioned that you're looking at the tolerability of both vehicles. Is that correct?
Yes, we are. Yeah, so there'll be vehicle mice for both the methylcellulose and the sodium citrate.
Yeah, I mean, just thinking about it, I agree completely about the PK, but if they're both well tolerated, then we'd rather go with the better formulation, as long as the PK is where we need it to be. Right? Assuming that. Indeed. So is that something, who would do that, Peter? I'm just wondering
So
about who would look at the difference in the formulation.
I'm not sure if Ahmed is on the line, but we have had those discussions to run a quick PK with these formulations.
Oh, you don't. Mm-hmm. What you see? What is something?
So yeah, that could.
Yeah, he is online. At least I see him.
The last study we had done with Chris Jones.
I'm sorry. Can I? Yeah, sorry, I couldn't unmute myself. I'm really sorry. Yes, we are planning to do the rapid PK with the new formulation, citrate formulation. It's going to be a 5-point PK, just the plasma concentration that's planned, and we are going to do it in early February. If that's okay.
That's good. I mean, in the ideal world, I mean, it doesn't matter where it's done, but in the ideal world, you would do it in the same laboratory where you're going to do your formulation or your pharmacology studies. But that's not always practicable, depending on what capabilities and licensing they have in those laboratories. So you may have to bridge to a PK study done in a different laboratory, but that doesn't matter. We just need to understand really, do we see exposures somewhere at least as good as what we saw with the previous formulation, and that we're going to have some decent exposures to go into the pharmacology study with. I think so. It's good to know we haven't that, even if it's that like PK light with 5 time points. That's fine. That's fine. That's good.
The study we had done with Chris Jones in the past with 2009 was done in the 0.5% CMC, and so that we had seen some efficacy, and that was well tolerated for that study, that vehicle.
Yeah, I think in an ideal world, if 2281 had been soluble in that, then we wouldn't need to have this discussion as much.
Mm-hmm. Yeah.
Going back to that reference I told you about, they've only reported 2 weeks dosing in rats, and it was well tolerated at 50 millimolar in rats.
Yeah, I believe that's
yeah.
If I recall, it's the same reference that we looked at when we were trying to, rapidly come up with something that we could test.
Yeah, I mean that paper still remains a sort of gold or a sort of a reference standard. For our formulation guys, they would always refer to that as well. So it's still relevant, that paper, despite the age. Okay, I think we've got the plan. We've got the PK coming. So that sort of addressed the question I had really. So
all right. No, I think that's a great point. So thanks for bringing that up to discuss.
Yep. Okay.
So now I'll move along to some recent data that we just received. So this work was done at Pharmaron, and I have some historical data of Charles River results for 2009 as comparison. I mentioned we sent 2009 along as a benchmark in this study, and I think at least the take home is that we're seeing double-digit micromolar activity in the hERG channel, the calcium channel, and the sodium channel for 2308. And I guess you could make the argument that it's slightly less active than 2009. But I think overall the take home is that at least in the limited assays that we've run with these lead compounds, that we don't see any red flags for cardiac liability. Some recent kinase data. These two assays were run at Reaction Biology. So it's really just to kind of finish off the data around some different ALK isoforms. And we haven't run ALK1, ALK4, ALK6 for all compounds, but looking back through, I think the trends remain the same with 2308. We see fairly equipotent activity, ALK1 and ALK2, and about a 8 to 10-fold less activity for ALK4, and about a 5-fold difference for ALK6. And I think this carries through with 2009 as well. But was really nice, I think, to see is that in this nanobread assay with these 4 ALK2 mutant strains in or ACVR1 mutants in the HEC293 cell line, 2308 is very active. I mean, it basically bottoms out their assay, and it certainly holds with the trend that it is anywhere from 5 to 10-fold more active than 2009. Any comments or questions about
Peter,
this new data?
What is the APY69? What's that?
That's just the reference, basically the reference compound for that assay. I just put that in there. So
very nice.
So they couldn't, I mean, couldn't they shift the assay to get hard numbers in the nanobread?
I mean, we could ask them. I don't. Is that something that's of interest? I mean, once you're getting down to that level of potency, I guess we did have some discussion last week or last meeting with Divya about what data to use in the target coverage, and I think, you know, at least with this, maybe the numbers change slightly, but I don't think the message changes that much.
I mean, it was more like getting at selectivity. I mean, so, I mean, it's good if you have absolute numbers to really put selectivity into context.
Mm-hmm.
So, you know, I mean, if you have something like ALK2 is 4 nanomolar, and then you have ALK1 here is like less than 3, you know, but when you have hard numbers, if you could get them, particularly in the nanobread assay, I was more intrigued by whether or not you can get full curves out of those and then report, you know, more meaningful selectivity numbers. Oh yeah, but as I say.
Yeah, I mean, certainly I can go back to them and see what, you know, if it's possible for them to go lower. I'm not.
That may be a problem too. So
yeah,
it's not a big deal. Yeah.
Okay, well, I'll make a note of that and see if that's possible. I think this can run through this pretty quick. This is tables that we've seen a number of times, but just highlighting some of the new data that's just come out. I did find a mistake here on the previous slide, so I've updated this with actual 2308 data. I think it was carried over for the last few months. The data for this ALK5 and fold over the nanobret was actually 2306, so this is actually a little bit better. And just again finishing out this table with some new data for 2308. You'll see that there is still some data gaps for 2281, so we'll work towards filling out those tables. And essentially that's what I'm going to discuss next. So for 2281, in order to finish off that, all the data that's missing at the moment, we'll need to run the kinase panel and safety 44 panel, as well as CYP inhibition, aims, and dose linearity. We have efficacy data from the FOP model upcoming, so that'll be interesting. And then having PK data for these 3 compounds from the mice in those studies. Rebecca discussed the dose tolerability. So that's will be completed in the next few weeks, and we discussed that we're putting those quickly into as quickly as possible into the queue for DIPG animal models. Now, I think once we have some the data from the FOP, if we're comfortable that we see some efficacy with 2281 and 2308, I think, you know, I'm leaning more towards 2281 likely being the development candidate. We're certainly going to need more of this material soon, relatively soon, particularly if we want to go into some more studies with this compound. So I propose that we think about doing some scale up and root scouting for 2281, and you know, at least in the early steps of that synthesis, it overlaps with 2308 anyway. I put together this timeline, and I've updated it a little bit based on some of the recent, I guess, delays in picking a development candidate, as well as ensuring that it matches up with some of the grants that we recently submitted. So here's where we are today. We're completing those data gaps, as I mentioned, and then we've started some of the efficacy studies with our 3 lead candidates. So I'm hopeful that once we start to see some of this data, and if we certainly start to see some efficacy in at least one of those DIPG animal models, we'll be in a position to pick a development candidate in the second quarter of this year. A lot of these other milestones, I think, have basically remain the same, but I have pushed out our target date for IND submission to the middle of 2027. Does anyone have any comments or questions on our timeline?
It's not so much in the timelines, but something to build in if we don't already have it. So if we progress 2281,
Yes.
we know that 2308 is effectively going to be an active metabolite. We certainly see it preclinically, and chances are you're going to see that in the clinic. So in terms of the sort of PK package, we would have to do all bells and whistles on 2281 and fully characterize that. But in vivo wise, we would be expected to deliver understanding of the intravenous pharmacokinetics of the active metabolite in the toxicology species. So there's an understanding of the likely distribution and clearance of the metabolite that could be predicted for human. So what that would mean is, subject to confirmation that rat and dog are suitable tox species from the metabolite ID work, we'd have to factor in some IVPK for 2308 in those species if we don't already have it. We've probably got it. I don't know if we have it in the rat, actually.
No, we don't.
No, so it's not a lot, but it's something you'd have to cost, compound supply. We'd have to have availability of 2308, even if it's not the preclinical candidate. We'll need to characterize it in some detail. In vitro assays, obviously, that's easier.
Yeah, and that
would be done. Yeah.
We wouldn't be talking tens of grams.
No, it's mgs. No, you're looking at 1 mg per kg, 3 rats, so 3 dogs. It's milligrams. It's not a lot. Yeah.
So we should.
That's a good point. I think we should do it across all the species. I mean, we had a project one time. I mean, the metabolite profile across the species had looked very different. I mean, the metabolites you were seeing in human that was not being seen in mouse and rat and stuff. So I think it's a good point. It's wise to kind of just take each species, mouse, rat, dog, and human, and just do a whole metabolite workup across them.
Yep. Now that's all planned out in this. Yeah.
That's an in vitro assay. So you do that in vitro in microsomes or hepatocytes or both.
Yeah.
And you're hoping to pick you hope that the 2 species you want to use show the same metabolites as human. Mm-hmm. And that's your way forwards there. So that is in the plans. We had that on the PK slides last time.
Yeah.
The recommendation was to get that cross-species MET-ID study on the candidate,
On the
it could be
candidate,
2281
right.
type thing, yeah.
So yeah, again, that's why things were pushed back slightly, just until we ensure that we're moving ahead with a compound we feel comfortable with, and then we'll put that into the metabolite ID study.
Thank you.
Okay, so that's essentially everything I had from my end, and I'm going to stop sharing my screen. And Dan, if you're ready, we can turn it over to you.
Sure thing, that sounds good. I'll share my screen now. Let me know if you can see that, everyone.
Looks good.
Nice one. Cool. So just for context, I gave a talk last week internally at Charles River, just updating on the ongoing ALK2 PROTAC work, and Kim thought it would be a good idea to do one this week with you guys and get your thoughts on what we've done and where to go next. So just kind of the background slide, which I think we've all seen before, talking about why ALK2 is interesting. It plays a critical role in the BMP signaling pathway by phosphorylating these receptor-regulated SMAD proteins, and that ultimately regulates gene expression via a SMAD158, SMAD4 signaling cascade. And ALK2 is an interesting target in DIPG, which we know, but also in FOP and anemia in myelofibrosis patients. And a selection of ALK2 inhibitors have been reported across different chemotypes. We have these blueprint type compounds like fidrisertib, which is for FOP. We have momelotinib, which is the non-selective kinase inhibitor for myelofibrosis, and also we've got compounds like M4K2009. And so kind of to develop our ALK2 degraders, we started off by taking these truncated ALK2 ligands and docking them within the ALK2 ATP binding pocket, and this kind of just showed the RR piperazine vectors extending out into solvent, and that's kind of how we designed our PROTAC. So we designed an initial set of about 15 compounds, and scientists here at Charles River synthesized them. And ultimately, all of these 15 compounds were screened using a Western blot screening approach in HEK293 cells that were transfected to express a hybrid-tagged ALK2 vector. And so essentially, we treated the HEK cells with each compound at a concentration of 1 micromolar for 24 hours, and then measured ALK2 hybrid expression. And really, from these studies, we identified a couple of interesting chemotypes, and probably two of the nicest representative compounds were this compound, compound 4, and compound 10. And what you can see is that the really nice thing from a hit finding perspective was that these two compounds were completely structurally distinct from each other. So one recruited VHL as the E3 ligase, while the other recruited cereblon. They both use completely different linkers and completely different ALK2 binding portions, which so this was really great from a hit perspective. As kind of part of our hit validation, we tested out all of these compounds in dose response Western blot studies. So the hit compounds were 3244 and 3140. So we did dose response Western blot experiments going from 30 nanomolar to 3 micromolar, and saw nice dose responses for both of these compounds in that hybrid tagged ALK2 system. And then, as well as that, we included a kind of a negative control PROTAC in these hit validation studies. This was a compound 3118, which didn't show any ALK2 degradation in our initial screening, and also when we took the concentrations up from 30 nanomolar to 3 micromolar, we didn't see any ALK2 degradation. So this was a nice kind of control. Then we've got the nanobret ALK2 IC50 data generated at UNC by Stephanie, which was really nice. We got that through recently, and that just confirmed in-cell target engagement by all of the compounds. And we've also got the experimental physchem and the in vitro ADME data for these compounds. And essentially, the key thing to take away here is that 3244 had kind of really nice metabolic stability, but wasn't very soluble in our fed state simulated intestinal fluid. And 3140 was the opposite. We had really nice solubility, but because of a PEG linker, the compound wasn't metabolically stable. And so, kind of moving into our hit expansion phase, we decided to focus on the cereblon-based PROTAC 3140, and we synthesized another small number of compounds, essentially where we wanted to improve the ALK2 degradation potency and also the metabolic stability. And our method to do this was to replace the PEG linker with a load of saturated and heterocycles. And we also included a few different cereblon ligand as well. That was based on a phenyl dihydrouracil structural motif. And again, we profiled these compounds using the Western blot screening method. So we dosed each compound in HEK cells at a concentration of 1 micromolar and then measured ALK2 degradation by Western blot. And then on these two plots, you can see the percentage ALK2 degradation at 1 micromolar plotted against either the metabolic stability in human liver mics or the kinetic solubility in FESIF. And the goal was to identify more potent compounds with better metabolic stability. And one of the compounds we identified which achieved this was this one shown at the top, where we've got an improvement in both of these things. And we also got adequate solubility. And so we termed this compound M4K2208 and took it through into further in vitro profiling. We did our dose response Western blot experiments going all the way from 1 nanomolar up to 30 micromolar. And you can see from the dose response Western blot, we got a really nice dose response up to about 1 micromolar, where we achieve about 65% ALK2 degradation, at which point the degradation kind of plateaus. And this is indicative of a hook-like effect, which we see with protein degraders sometimes, and it's something you need to optimize out. We did a time course just to show that ALK2 degradation was a time-dependent process. And again, we've got the nanobot data that was generated by Stephanie at UNC. And then at the bottom, we just have the kind of the in vitro admin experimental phys-chem properties compared with the parent compound, M4K3140. So this was quite nice. And this was a nice promising initial profile. So we took this compound through into selectivity studies, and we kind of used two methods to investigate the selectivity of this compound. First of all, on an unbiased selectivity perspective, we used quantitative proteomics, mass spec-based proteomics, in 2 cell lines. One was ACVR1 transfected HEK293 cells, and the other was these U87 glioblastoma cells, which endogenously expresses ACVR1. And you can see in both of these cell lines, we get a significant reduction in ACVR1, which was great, recapitulating the Western blot results. And we identify a selection of off-targets, and we've kind of got a rationale for each of the off-targets that we see here. And probably the most interesting one, because it shows up in both of the volcano plots, is this protein PDE6 delta. And we think that PDE6 delta, we know it contains a glycine-rich loop, which we know is a degron motif that can be identified by imide-based molecular glues, and we think that 3233 could be binding to cereblon, changing the protein surface and enabling an interaction to be formed with PDE6 delta, leading to neo-substrate degradation. That's the rationale we've kind of got for that protein. We also wanted to look at the selectivity of a closely related protein ALK5, and we know that ALK5 inhibition gives you a lot of negative effects. And so working with Stephanie again, we did the ALK5 nanobret assay, and in this assay, Stephanie included the ALK2 and ALK5 selective inhibitors, London 212854 and galinisative. And just what we can see here is up to a concentration of 30 micromolar, we don't get any ALK5 inhibition. And also by looking at our U87 proteomic data, we can see that we don't get any reduction in endogenous ALK5 expression. So that was nice selectivity data. Because this compound had a nice profile, we decided to use it as kind of a chemical probe to investigate the mechanistic basis for ALK2 degradation. And so we did a load of different competition experiments, which kind of well-established method of protein degradation by PROTACs is you get recruitment of your protein of interest and your E3 ligase, in our case cereblon, and this leads to ubiquitination of the target protein and degradation via the proteasome. And so the first thing we did was we demonstrated that you do need to recruit both ALK2 and cereblon in order to get ALK2 degradation. And so we did that using competition experiments with ALK2 and cereblon binders. But we also did that by making the negative control compound 3233NC, which essentially is methylated on the glutaramide ring. And what you can see in the Western blot panel here in the third lane is we just don't get any ALK2 degradation with that compound, which was really nice to see. And we also got nanobot binding data on that compound to show that it bound with essentially similar affinity to the parent compound. We then looked at the role of a proteasome and a lysosome on ALK2 degradation, and this is where we got some interesting results. And we found that essentially when we blocked recruitment of a proteasome, the action of a proteasome or the lysosome using MG132 or bifilomycin, individually, we didn't get a complete rescue in ALK2 degradation. We kind of got some rescue, but by no means complete. And that suggested there could be a role of both of the proteasome and the lysosome in ALK2 degradation. And so we then did a third experiment where we treated these HEK cells with the degrader as well as MG1321 bifilomycin, and you can see we get complete rescue of ALK2 degradation when we do that. And so that kind of suggested that the mechanism of degradation was going via a cereblon-mediated ubiquitin proteasome lysosome combination, and kind of that's been demonstrated before. It's not common, but it's been demonstrated before for other transmembrane kinases like EGFR. So as part of the next stage of optimization, we wanted to particularly look at kind of protein degradation as a tool for DIPG, but also for glioblastoma, because we know that ALK2, some glioblastoma cells are sensitive to ALK2 inhibition. And in order to try and improve the brain penetration of our potential compounds, we knew that if you removed the methoxy group in the center of this phenyl ring and replaced it with a fluorine, in the ALK2 inhibitors, you got improved brain to plasma concentrations. And that's what we set out to do. And so we synthesized another half a dozen compounds with this modification in, including M4K2250, which is just a matched molecular pair of 3233. And then when we profiled this compound, we were really pleased to see we got a much better degradation profile. We saw with the parent compound, we got plateauing degradation at about 60%. With this compound, we got pretty much a complete degradation, and we never saw a hook effect, which was really nice to see. And then we've also got the in vitro ADME data for this compound, and essentially we see that when we replaced the methoxy with a fluorine, we got an improvement in macrosomal stability in human mice, but we did lose some of the solubility. And so we next wanted to investigate the phenotypic effects of our PROTACs. And so we started off by using an in-house cell viability assay in U87 glioblastoma cells, which have been reported to be sensitive to ALK2 inhibition. And so in these studies, we used both of our most promising PROTACs, which were M4K2233 and 3250, which are the red lines in both of these plots. For the ALK2 inhibitors, we used the parent ALK2 inhibitors M4K2009 and M4K2163, which are the green lines in each plot. And what we can see here is that when we treat these U87 cells with the ALK2 inhibitors, we get a small decrease in cell viability, which to a level which is consistent with what we see in the literature. And that's what we see here. When we treat with our PROTACs, we get a much more potent cytotoxic effect, which was really great to see. But then we did a number of control experiments to look at, to confirm that you did need to recruit the E3 ligase cereblon to get protein degradation. And so we also tested out the negative control compound 3233. And we also did co-dosing experiments where we treated with the PROTAC in addition to a saturating concentration of pomalidomide, which binds to cereblon and would block the recruitment by the PROTAC. And what you can see from the blue and the purple lines in each of these graphs is that when we take away the ability of our PROTACs to recruit cereblon, we don't get any difference in cytotoxicity. And this was really strange to see because it indicates that the phenotypic effects we see are pretty much independent of a cereblon-based mechanism of action. And because we know that cereblon is required for ALK2 degradation, it suggests that the phenotypic effects we see could be independent of an ALK2-based mechanism of action, potentially. So that was really weird to see. And in order to see if it was just isolated to these U87 glioblastoma cells, we did the studies with Rebecca at the OICR, and these were preliminary N of 2 experiments in 2 of the patient-derived DIPG cell lines. The method was slightly different. Rebecca did a longer treatment, but the effects are essentially the same. We see that ALK2 inhibition is cytotoxic. We see that the ALK2 degraders are more cytotoxic than the ALK2 inhibitors, but we see that's independent of the ability to recruit cereblon because the curves essentially overlap, which is again suggesting that the phenotype we see is independent of ALK2 degradation, which is really surprising and a bit confusing. And so that's kind of where we are now. And so to summarize what we've done, we've developed these ALK2 degraders and done successive rounds of optimization for potency and in vitro ADME. We've investigated the selectivity and the mechanism of ALK2 degradation, and we've evaluated the phenotypic effects by doing CTG assays in different ALK2 sensitive cancer cell lines. But because of these confusing phenotypic results, we've decided to pause the development of these cereblon recruiting PROTACs until we can kind of get a better idea of what might be happening here. And so any thoughts anyone has would be really welcome. Kind of as the next stage, what we've done is we've initiated cell viability assays in the U87 cell line using our alternative hit compound M4K2244. And this compound is completely structurally distinct from our other more developed PROTACs. And so essentially we want to see if we get the same confounding phenotypic effects. If we get the same confounding phenotypic effects, then it suggests that the effects we see are independent of what the compounds look like. It's not something in the compounds that are giving us the cytotox, at which point we'll need to assess looking at a new target or moving on to something slightly different. And kind of that's all of the slides I have. And so I'd love to hear some thoughts or hear some questions.
Thanks, Dan. That was great. I have one question, and it started at slide 6, but you kind of answered it along the way with 3250 and the not having the hook effect when you actually replace the methoxy with the fluorine. Is there a rationale or a thought of why that happens when you add in that fluorine, or is it just what you see from the assay? I'm just curious if there's a way to like you mentioned, modify the compound to take out that hook effect.
Sure. So no, we don't have an answer to that. You could suggest some rationales, but they kind of all fall down at different points. It could be potentially due to better cell permeability, potentially, but the nanobret data would kind of suggest otherwise. That really, we think a hook effect arrives because you've not got a cooperative ternary complex formed, and so you would expect that because we've not got a hook effect in 3250, we're forming a more productive ternary complex. But the fluorine group is so far away from any of the kind of the protein-protein interactions, I'm not sure how that really would affect the productivity, the cooperativity of a ternary complex. So yeah, a lot of ideas, but
yeah,
really
no,
sure.
that's why it was kind of surprising to me that just that one change makes a pretty significant difference in your assay.
Sure.
Remind me again, what was the plateau for the other compound? Because this one only got down to 60.
Yeah, the better one. Yeah, it was pretty much complete, kind of 95, 97%.
Okay, interesting. My other question is, what's the turnaround of ALK2? So, like, I mean, if you degrade, does it come back? Does the half-life of ALK2 is established?
We're not entirely sure because you can't get antibodies for ALK2 on its own. We don't really know what the half-life of endogenous ALK2 is. You can do experiments with tagged stuff, with hybrid stuff, and I think Alex might have done some work on his tagged stuff, and it's maybe about 8 or 10 hours. But again, it's all tagged exogenous ALK2, so we can't exactly be sure on that.
Yeah, I'm not sure. Daniel, I mean, are you interested in looking at other phenotypes? So some of the historical data suggests 2 effects of ALK2, maybe one on, seems some proliferation effect, but maybe it's not all or, everything, but also effects on stemness and differentiation. So I'm wondering if you can test if your PROTACs have an effect on stemness properties.
That would be interesting to see if we can tie it to a phenotypic effect and see if it is different depending on the ALK2 degradation effect. That could be a good thing to do. Obviously, the viability effect was very easy to check, so we can just use a CTG assay, so that required very little resource, whereas looking at other stuff might be more complex, but that could be a good avenue to look down. Yeah.
Could you also comment on, so we're just looking at ALK2 degradation, but if these ligands bind to the other ALKs, I mean, have you contemplated looking at the kinetics of degradation of those ones? I mean,
the other ones, yeah, we could. So obviously ALK1 is the other kinase that we know that the ALK2 inhibitors bind pretty tightly to, and it would be really interesting to look at, say, ALK1 degradation or ALK4 or ALK6 and see if you even get improved selectivity there, because you're forming a ternary complex. That would be something that'd be really interesting to see. from a biology perspective.
So what does the genetic data say about these cell lines when you knock down ALK2?
Which cell lines? The DIPG and the U87 ones.
Mm-hmm.
So the DIPG cells, we know that when you knock down ALK2, you get cytotoxic effect. The U87 cells, it was just a paper which showed that when you treat with an ALK2 inhibitor, DMH1, you get a reduction in cell viability. And so that's where we decided to base our in-house assay off. Knockdown, I'll need to have a look at the paper again.
Thank you.
There's been some work on ALK2 viability in prostate cancer models and also ovarian cancer models. So there might be 1 or 2 cell lines, prostate and ovarian, not all, but some.
Yeah.
Just in case they're useful.
Yeah, they definitely could be.
We've us and others have shown in some models that if you do CRISPR genome-wide CRISPR screens, you can get ALK2 as a hit, so you knock it out and it is lethal. And I've tried knocking it down in a couple of the DIPG lines, and generally what we see is that there's an effect to start with. So you see a reduction in cell viability when you initially knock down ALK2, but then the cells kind of pick back up again. And I'm not sure if that's because of inefficient knockdown or some kind of compensatory effect. But yeah, it's not quite as clear cut as you knock it out and the cells die.
Okay.
Thank you.
I did have another question, Dan. Nice data. So is the thinking, because they're based on the M4K2009 and 2163 and they're more potent, is the thinking that maybe there's off-target effects that when you hit them just with a small molecule, it doesn't really do very much, but when you degrade the off-target effects you're having, that's what's getting that shift, rather than it being ALK2 dependent, it's off-target dependent, and the degradation is more important in that context?
That's a really great idea of being related to the off-targets, but it has to be independent of degradation, because we're taking away the ability to degrade via cereblon route. You could get degradation via a different route that we're not seeing, but it wouldn't be degradation of the off-targets.
Right, got you.
Yeah. We've done some proteomic work on the negative control compound, and we've kind of seen some proteins which are either upregulated or downregulated when you treat with both the PROTAC and the negative control, but we've not done any more work or had any more thoughts about that.
Yeah, I've got some more data that I can send you, by the way.
That'd be excellent.
More lines, pretty much the same story, but I can send that.
Perfect, thanks.
Okay, everyone. I think, is there any more questions for Dan? Well, I think we can probably wrap up now. So I appreciate everyone for coming, and we'll see you next time.
So just to check, sorry, Peter, the next meeting is in March.
Will be
is that correct?
in March. That's correct.
Yeah. Okay. Thanks.
All right. Bye, everyone.
Bye.
Bye.